My name is Ghada. I am from Egypt, but right now I'm working in Al University. I did this research in KSA, Mecca. Our talk today about molecular evaluation of idexx paratuberculosis screening ELISA test as a rapid tools for detection of Johne's disease among suspected small animals. Johne's disease or paratuberculosis is a chronic, progressive enteric disease of ruminants. It's caused by infection with Mycobacterium avium. Oh, okay. Thank you. It's characterized by therapy-resistant diarrhea and weight loss. As the disease represents a great problem among ruminant animals in Saudi Arabia, and due to its zoonotic possibility, we did this study. This study aimed to utilize molecular tools to evaluate the reliability of idexx paratuberculosis screening ELISA test versus traditional microscopical examination of acid-fast bacilli smears for rapid detection of Johne's disease among clinically suspected small ruminant in Mecca region, KSA. For this purpose, we'll use 3 different genetic targets, including 16S, rDNA, IS900, and IGS, were used for molecular diagnosis as a golden standard for the evaluation of the study. A total of 2,660 local sheep Naomi breeds were investigated in this current study during the period of November 2030 to June 2040 for suspected signs of Johne's disease. These animals belong to El Kahkaya slaughterhouse. This is the main slaughterhouse in Mecca, as well as 5 small farms around the area of Mecca. Clinically suspected animals were selected and isolated based on the history and clinical examination. Inclusion criteria: the animal should have depletion, emaciation with unresponsive to dewormed and antibiotic, intermittent diarrhea or softness, pasty stool, low-grade fever, lethargy, and depression. In spite of weight loss, the animals still have a good appetite. Intermandibular edema is very characteristic. Our investigation of 2,660 animals, we found 41 animals were selected at the beginning, suspected that they are infected with Johne's disease based on the symptoms. From these suspected animals, fecal sample and blood sample were collected from all the animals. Fecal sample were subject for both conventional microscopical examination and the molecular examination. Blood sample were used for serum separation, then condition of immunological assay using idexx paratuberculosis screening of ELISA. First, we do the conventional microscopical examination. Collected sample from clinically suspected cases were initially evaluated by the microscope for acid-fast bacilli. For this purpose, we used the Nelson stain. Then we do ELISA using Mycobacterium paratuberculosis antibody test kits, which is idexx laboratory. According to manufacturing structure, we do the same. We didn't do any modification. All assays were run in duplicate. Any assay with in between coefficient of variation of 10% were repeated, and the second result were used for the data analysis. Molecular detection and identification: DNA extraction from the fecal samples. DNA was extracted from suspected animals. using Bromega kits. Amplification of selected genome target. For this, we use 3 different genomes, targeting the amplification from DNA obtained from all suspected animals. This includes 16 srDNA, IGS, and IS900. The hyperviability 500 base pair segment of 16 srDNA was amplified using the previous described universal published by Hull 2003. The hyperviability region of the integrated spacer IGS was amplified and suspected to the sample using the primer ITS A1 and ITS A6 published by Muhammad 2005. Finally, the IS900 was amplified using the primary set and published by Kerr 2004. All primer sets were obtained from IDT intelligent DNA technology. Then S sequence analysis. Our results. As you can see in this picture, this initially the microscopical examination, and we have by the error in front of the error we have the asfas stained by Zen-Nelson. Out of the 41 sample we have examined with symptom of Johne's disease, we found 14 sample positive with Zen-Nelson. We do the ELISA test and we found 15 out of the 41 positive with ELISA. So we do the molecular test. And first, this is a representative gel. It's not all the sample. The L lane is our base pair leader. And as you can see, it's 500 base pair sample from 1 to 9 are positive. Sample 10 and 11 are negative. 12 is the negative control. And regard this, we found, we detect that we found 26 of the 41 are positive for 16 srDNA target. So we do more. Out of this 16 srDNA positive sample, only 18 were positive for IGS. As you can see, this is a positive sample, this negative sample, and 12 is a negative control. So, which is which? Further investigation of the selected 18 sample was confirming Mycobacterium species infection using both sequencing analysis, obtaining IGS targeting amplification as well as amplification of the IS900 target has confirmed the presence of Mycobacterium avium in only 14 samples. So, let's return from this big mass. We have the table here. By microscope, we found 14 sample positive, okay? By ELISA, we found 15 sample positive. When we do the molecular biology, at the beginning, for 16S, we found 26 positive. IGS, 18 out of this 26. 14 positive for IS900 and 14 positive for when we resequencing. Yeah, so make your life more complicated, which is which. Not all the...
Excuse me, can you use the pointer so we can...
Oh, it's okay. Oh, skip this. Okay, now, Finho. It's not here. Oh, okay. It's okay. It's okay. So, ما فيش حل غير كده. Okay. So, how many we have true positive and how many true negative? We have 7 samples true positive by the microscope, 12 with ELISA. So this is good news. ELISA is good. True negative, we have 7 with the microscope, 2 with ELISA. Still good. True negative, 18, 24, false positive, 7 and 3. So let's make it our life easy and make it by percentage. We found that by ELISA, comparing to microscope, as our background, or regarding to our molecular results, we found that ELISA are high sensitivity, where there is a sensitivity is 78%, plus 50% with microscope. Specificity, 88%, was 70%. Positive predictive value, 80. Negative predictive value, 92. So, in conclusion of this study, reveals that the feasibility of idexx paratuberculosis screening ELISA as a reliable tool for rapid detection of Johne's disease among suspected cases of small ruminant, which would support the control program of Johne's disease among commercially small ruminant herbs in Saudi Arabia. That's it. Thank you.
Thank you.